Welcome to this video on glycogenesis. This is an important energy metabolic pathway in which body stores energy fuel for use during fasting and sleeping. We will explore the sites where it occurs, its detailed mechanism, key enzymes, regulation, and overall significance. Let us explore the organs and sites of glycogenesis. Glycogenesis is the enzymatic synthesis of glycogen using glucose. The liver and skeletal muscle are the two primary organs of glycogenesis, which stores about 100 gram and 400 gram of glycogen respectively. Other cells and organs, including brain astrocyte, also stores a small amount of glycogen. To understand the mechanism of glycogenesis, it is important that we explore the basic structural elements, including branching points and types of bonds in a glycogen molecule. Glycogen is basically a homopolymer of alpha-D-glucose units, which are linearly connected with alpha-1,4 glycosidic bonds, shown here with red lines. And the branching point is formed by alpha-1,6 glycosidic bond, which is shown here with the blue line. How these bonds are formed will be discussed in detail during the mechanism of glycogenesis. Before discussing the mechanism of glycogen synthesis or glycogenesis, let us review the enzymes which are involved in this process. There are 6 total enzymes which are used in this process. First is glucokinase, active in liver, and hexokinase in rest other tissues. Second enzyme is phosphoglucomutase. Third one is UDP glucopyrophosphorylase. These three enzymes are used in the synthesis of UDP glucose, which is an activated precursor for the synthesis of glycogen. Fourth enzyme is glycogenine, which is used to synthesize primer. Primer is an absolute requirement for the synthesis of glycogen. And the fifth and sixth enzyme, glycogen synthase and branching enzyme, these are the core enzymes which are used in the synthesis of glycogen. We will discuss these in detail in the coming up slide. Let us discuss the mechanism of glycogen synthesis or glycogenesis. It completes in 4 steps. The synthesis of UDP glucose, which is the activated precursor for glycogen synthesis. Second is primer synthesis by glycogenine, and third is elongation, and the last is branching point. We will discuss these one by one in detail in the coming up slides. Let us examine the synthesis of UDP glucose. This completes in 3 steps utilizing 3 enzymes. The first step is conversion of glucose to glucose 6-phosphate at the expense of 1 ATP molecule by enzyme glucokinase, which is active in liver, and hexokinase, which is active in other cell type. Glucose 6-phosphate is converted into glucose 1-phosphate by mutase enzyme. Glucose 1-phosphate is finally converted into UDP glucose by UDP glucopyrophosphorylase utilizing 1 UTP molecule. UDP glucose is the activated precursor for the synthesis of glycogen. Let us discuss primer synthesis. Once UDP glucose is available, primer synthesis begins on the tyrosine residue of a protein known as glycogenine. This is a protein which has enzymatic activity, and it starts adding glucose unit one by one by forming alpha-1,4 glycosidic bonds. Once 8 to 12 glucose units are added, this small oligosaccharide segment serves as a primer for glycogen synthase to elongate the chain using UDP glucose. Therefore, glycogen synthase requires a primer. You should know the difference between glycogenine and the primer. Glycogenine basically synthesizes the primer. It is not the primer by itself. Let us discuss the third step, glycogen synthase activity or extending the primer. Glycogen synthase requires a primer which is attached to or synthesized by glycogenine and UDP glucose, which is the activated precursor for glycogen synthesis. Glycogen synthase starts extending the chain by connecting glucose moiety through alpha-1,4 glycosidic linkage. Once 12 to 14 glucose moieties are attached or added, the branching enzyme starts making a branch point, which we will discuss in the next slide. Let us discuss how branches are added in the glycogen molecule. Once the glycogen molecule has been extended, the chain has been extended by 12 to 14 glucose residue longer. Branching enzyme detaches a segment containing 6 to 8 glucose residues by breaking down alpha-1,4 glycosidic linkage and attach it to the existing chain or to the new chain by forming alpha-1,6 glycosidic bond. This alpha-1,6 glycosidic bond is the branch point, and these new branches will be then extended by glycogen synthase, and branching enzyme will keep on adding new branches. So these two enzymes will keep on working, and ultimately a glycogen molecule which is highly condensed and highly branched is formed. Let's review the significance of glycogenesis. Physiologically, it is very important because it maintains or prevents hyperglycemia by conserving glucose in the form of glycogen. In addition, it also maintains blood glucose level while we are fasting or sleeping for up to 18 hours. Clinically, it is important because the enzyme, when defective, lead to diseases called glycogen storage disease or GSD. The first one is GSD type 0, which is caused due to defect in the glycogen synthase enzyme. This is associated with hypoglycemia. And the second disease is GSD type 4, which is caused due to defect in branching enzyme. In this condition, the glycogen formed is defective. It tends to precipitate, causing liver damage and hypoglycemia. Glycogenesis is regulated by hormone insulin and glucagon through their action on glycogen synthase. For example, insulin activates glycogen synthase, thereby activating or inducing glycogenesis. On the other hand, glucagon inhibits glycogen synthase, thereby inhibiting glycogenesis. Thank you very much for watching this video. Please like it, share it, and subscribe it, and write comments. Until next time, you take care. Bye bye.